This scientific overview examines the biological rules that govern viral entry and organ susceptibility. This presentation is by Bryan Geniuson, founder, principal research scientist, and research systems architect at Natural Scientifics, an independent research platform dedicated to systems biology, disease mechanisms, and evidence-based scientific education. This discussion addresses how viruses initiate infection at the cellular level, how they gain access to host cells, and why early entry dynamics determine downstream tissue involvement and disease severity. Viral infection does not originate with clinical symptoms. It originates with cellular access. A virus is incapable of replication, dissemination, or pathogenic activity unless it successfully penetrates a host cell. Viral entry therefore represents the earliest and most decisive biological gate in the entire infection cascade. Viruses function as obligate intracellular systems. They lack autonomous metabolic and replicative machinery and must exploit host cellular structures to survive and propagate. To accomplish this, viral particles are structurally adapted to recognize and bind specific molecular features on host cell membranes. These molecular features, referred to as viral receptors, dictate which cell types and tissues are permissive to infection, a property known as viral tropism. At the surface of epithelial and endothelial tissues, viruses encounter receptors such as angiotensin-converting enzyme 2, cluster of differentiation 4, or sialic acid-containing glycans, depending on viral lineage. These receptors are not uniformly distributed throughout the body. Their expression varies according to tissue type, cellular differentiation state, developmental context, and physiological conditions. This heterogeneous receptor landscape is a primary determinant of organ-specific vulnerability. Receptor engagement anchors the virus to the host cell membrane, but is frequently insufficient to permit entry. Many viruses require proteolytic activation by host enzymes, proteases such as transmembrane serine protease, 2-cleave and prime viral surface proteins, inducing conformational rearrangements that enable membrane fusion or internalization. This process, known as protease priming, functions as a regulatory checkpoint governing viral entry competence. Once primed, viruses enter host cells through one of two dominant pathways. In direct membrane fusion, the viral envelope merges with the host cell membrane, releasing viral genetic material directly into the cytoplasm. In receptor-mediated endocytosis, viral particles are internalized within vesicular compartments, where endosomal acidification and enzymatic activity facilitate membrane fusion and genome release. Failure to escape this compartment results in viral degradation. Following entry, viral genetic material is released into the cytoplasm or transported to the nucleus, depending on viral genome type and replication strategy. This intracellular routing determines replication kinetics, host resource exploitation, immune detection timing, and the activation of cellular stress and inflammatory pathways. Crucially, viral entry dynamics are not uniform across the organism. Distinct tissues express unique combinations of receptors, proteases, and intracellular cofactors. This explains why respiratory viruses preferentially infect pulmonary epithelium, why certain viruses invade the central nervous system, and why others target vascular endothelium or gastrointestinal mucosa. In the lungs, viral entry into airway epithelial cells disrupts barrier integrity and drives inflammatory respiratory disease. In the brain, entry across neural or vascular interfaces facilitates neuroinvasion and neuroinflammatory injury. In the vasculature, endothelial infection promotes vascular dysfunction, inflammatory signaling, and tissue damage. In the gastrointestinal tract, viral entry alters epithelial permeability and immune homeostasis. Across viral diseases, a consistent systems-level relationship emerges. Entry efficiency determines replication capacity governs tissue injury, and tissue injury defines clinical disease expression. This cascade explains why early containment of viral entry can profoundly influence disease trajectory and why entry-targeted interventions remain a central focus of antiviral strategy. Understanding viral entry and tissue tropism provides a mechanistic foundation for interpreting disease patterns, anticipating organ involvement, and designing preventative and therapeutic approaches. It also establishes the biological context necessary to understand how immune surveillance is activated, how it may be evaded, and how immune responses themselves can contribute to pathology. In the next installment, this series will examine how the immune system detects viral invasion, how viruses disrupt immune signaling pathways, and how immune dysregulation can become a driver of disease progression. The complete scientific literature and extended reference materials supporting this presentation are available separately by purchase. This content is provided for educational and scientific discussion purposes only and does not constitute medical or therapeutic advice.